To the final day. So this session is exercise and cancer. We have 4 speakers. Jim Carson from University of Tennessee will be speaking first. His talk will be remote fed in. He was unable to be here in person. I'll be speaking second, then Mike Lisio from University of Ottawa will be speaking third, and Talbert from the University of Iowa will be speaking last. So without further ado, we'll get started, if we could please, with the first speaker, which is Jim Carson from the University of Tennessee.
Good morning. I'd like to thank the organizers of the International Biochemistry of Exercise Conference for allowing me to present, and also for their graciousness as I'm recovering from some long-term orthopedic issues successfully and allowing this to go on. So I'm going to speak to you today on disrupted muscle metabolism in cancer and treatment, a role for exercise. There are no conflicts in the work presented today has been funded by the National Cancer Institute. It's going to highlight some work from Jessica Halley, currently in my lab, and Brittany Counts-Franck, who recently graduated, is now working with a very strong pancreatic cancer and cachexia group in Indianapolis. So cancer patients in most preclinical models of cancer demonstrate decreased physical activity and increased fatigue. We really don't have a sufficient understanding of how physical activity impacts cancer-induced metabolic dysfunction and aberrant protein turnover. So today I'm going to describe to you a little bit about the role for disuse and inactivity and make a case for that. I'm going to talk about the potential for increased physical activity in response to contraction as an important aspect of skeletal muscle during cancer. And then I'm going to finish with some current work we're doing with cancer treatment's role in looking at chemotherapy and long-term muscle dysfunction. So there's strong agreement, and it's very nicely shown here, that loss of lean body mass contributes to morbidity and mortality with a host of problems, as you can see, related to immune function, wound healing. There's weakness, infection, and ultimately pneumonia and increased risk of death. And there's tremendous interest then of how to stave off this loss of muscle to protect against these problems. So within cancer, and this is very nicely shown in this recent review, aspects have really focused on tumor growth and tumor-derived factors and tumor-derived changes to the systemic environment that could cause alterations in muscle and now adipose tissue and others. And there's a growing interest in trying to determine how chemotherapy, as most patients are being treated, contributes to these changes. And I like this review because it also brings in the idea that cancer patients are normally inactive, and inactivity could have another role. Things that are often examined as drivers of this muscle loss included listed here: inflammation and related signaling, mitochondrial damage, hypercatabolism leading to accelerated degradation of protein, and suppression of protein synthesis, as well as with moving into myogenic responses related to satellite cells and others. But what I've noticed over the 30 plus years I've been involved in the muscle plasticity field is that currently, especially in some of the things with cachexia and wasting, the current investigations heavily emphasize the upstream regulation of the cancer disruptions to oftentimes mainly muscle mass, contractile function, but we really don't have a very good understanding of the downstream health effects that link muscle to the cancer survival. So why does muscle mass maintenance promote health of cancer patient? I think it's really critical. We're really going to address and get effective therapies for this condition. And to our early induced changes in systemic and muscle metabolism that often occur before cachexia, negatively impacting or driving downward spiral of health. So I had the good fortune to work on the faculty at the University of South Carolina with Dr. Steve Blair, and actually served as chair for a number of years and had a lot of interesting discussions with him. And like any outside-the-box thinker, he's often controversial, but I saw some commonalities related to his being involved in obesity and health, but really working against the idea that it was all about mass, fat mass, as a matter of fact, but there being an obesity paradox that he's carefully outlined in many papers, that oftentimes if you stratify by fitness level or other risk factor modulators, the risk of being obese will be reduced. So I began to wonder, with cancer wasting or others, is it one size fits all? Because often we're talking just about muscle mass, loss of muscle mass. But is there potential that there's other things going on with the muscle that might be a determiner of health? So there's certainly a role for muscle in systemic metabolism, and I don't have to tell this audience that. And when we look at metabolism as far as glucose metabolism in the fed and fasted state and the potential role of muscle has in those two states, it becomes critical. And then as we get into a malnourished state, or maybe a better term for the cancer talk, a catabolic state, we can see skeletal muscle has a strong rationale for being involved in health because it will contribute to tissue structure, function, immune responses, neural function. So I think when we talk about treatments that might not allow muscle to do this, I think we might be going down a dangerous path. But there's importance in also looking at the systemic metabolic flexibility in health, and skeletal muscle has a key role in this. I've learned a lot of what I know here from Very Good Pasture when he diagrams this. This diagram nicely shows the role of this metabolic flexibility of going from inactive to active, from going from eating to fasting, and the changes that occur when that happens in muscle related to fatty acid oxidation, glucose oxidation, glycogen storage, and that also targets fat and lipolysis. So this has begun to be a knowledge in working with cancer and cancer cachexia and systemic metabolic dysfunction with cancer, and nicely detailed by Teresa Zimmer's group in pancreatic cancer patients, is the interaction between adipose tissue, muscle, and the tumor factors. I think you could also put the liver in here as a critical mediator. And then also work detailing how the tumor could target osteokines from bone and myokines, which have big effects on metabolism and disrupt them, and then lead to also metabolic and health disruptions. So when we look at preclinical models of cancer cachexia, it's clear in some of the most established models, including the colon 26 model, shown some time ago by Dr. Gordon Lynch, or the Lewis Sloan carcinoma model. I'll give some examples today. That's nicely shown by Bruce Spiegelman's lab and others. And we've shown in the APC-Min that there are metabolic disruptions in these preclinical models to systemic metabolism. A lot of that having to do with metabolic flexibility, and it's not just hypercatabolism, although that's been reported. And so what's less clear when we look at some of these is when this is occurring. Is this potentially a driver or an early onset phenomena? And so I began to look at then with this role for disuse and inactivity. And I was very lucky to have a postdoctoral training in the lab of Dr. Frank Booth. And while I was there, I worked with a fellow with Dr. Mark Hamilton. And they were just beginning to form their hypothesis related to Frank's work with NASA and muscle disuse and lipoprotein lipase activity into what will become the sedentary behavior SEDS, emphasis on time sitting today, and metabolic dysfunction. And I really kept this in the back of my mind when I'm looking at a lot of what we know with the metabolic disruptions and others with cancer, which I won't present today, but where cancer patients and others are highly inactive. Inactivity is certainly a risk factor for sarcopenia. It's been acknowledged now, and when we often see inflammation, aging, and it can often accompany many of these other disorders, it's interesting to speculate whether inactivity would make you more susceptible to the negative effects of inflammation. Susan Kandarian and Bob Jackman nicely showed years ago that with disuse or immobilization or inactivity, a lot of the catabolic pathways induced in muscle that negatively affect protein synthesis or elevate protein degradation are very similar to what we report in many of these other cancer-inducing catabolic environments. So years ago, Kristen Volk-Alvis in my lab, we began to work on the APC-Min mouse, a genetic model of cancer cachexia. The Min stands for multiple intestinal neoplasia. It can be related to colon cancer. But we started to use this long window of the cachexia development from 13 to 19 weeks of age or longer. And in one of our experiments, as we kept a large cohort of these mice housed in wheel cages, and followed them out. And because it is a genetic model, we got a variety of cachexia between the mice. But when we went back and looked at the data, the mice that developed the severe cachexia at the end of our study started to show diminished wheel activity and volitional wheel activity long before they were showing signs of cachexia or body weight loss. And this led us to really be thinking about, is a reduction in physical activity contributing to the initiation and progression of cachexia? Brittany Counts in my lab extended some of this work by looking at cage activity measurements in the APC-men mouse. And what we found is that when we divide the mice up to either pre-cachexia or cachectic mice, we see that the cachectic mice that will develop cachexia have considerably less cage activity. And when we correlate that, we can see that reduced cage activity is directly related to greater body weight loss. So we decided to extend these studies, and Brittany went in and used the LLC model. You can see it diagrammed over here where it's more not a genetic model, but where we inject LLC cells in the flank and follow them for approximately 4 weeks. It's a very well characterized cachexia model. When we examine tumor volume, we see that really out to 10, 12 days, there's really no palpable tumor, and the tumor starts to be palpable and start to have measurable growth somewhere around 2 weeks. During the 2-week period. So what we decided to do before these animals developed cachexia, before the tumor was large, we put the mice in clams metabolic cages between days 15 and 20 and monitored them, and then followed them out the rest of the study. One thing we try to take advantage of this in our hands, at least when we do a large cohort of animals, we see quite a variety of tumor mass at the end of the study, and also the tumor growth rate during the study. So when we measured physical activity levels in these mice in the clams cages between days 15 and 20 before developing cachexia, and then followed them out, we saw that cage activity early on was related to tumor mass at the end of the study, being that a large tumor mass at the end of the study was associated with low cage activity early on. And additionally, low cage activity early is also associated with low hind limb muscle mass at the end of the study. And interestingly, cage activity was also inversely related to IL-6 levels circulating at the end of the study. So related to metabolic response, what we found early on before the development of cachexia and the really development of large tumors, we found that there were disruptions to lipid oxidation before cachexia development, and higher lipid oxidation in the tumor-bearing mice. And this higher lipid oxidation was associated with lower muscle mass at the end of the study. And interestingly, when we took the animals and divided them in cohorts of animals that had low growth small tumors versus high growth large tumors, we saw an increased rate of lipid oxidation early on in the mice that would go on to develop high growth large tumors. So we wanted to extend these studies because we know that the anabolic response of muscle is really dependent on fasting, feeding, and activity. And I've learned a lot of this from Dr. Phillips. And this very nice article shows that how even disuse can affect this relationship between feeding and fasting and a muscle anabolic response. So we've had a research done again that Brittany did in my lab with the LLC tumor implantation model, and then we followed a fasting feeding paradigm that closely mirrored what Scott Kibble has done very nicely over the years, and Brad Gordon has also published, where we fasted for 12 hours, where we allowed a subset of those to have a pellet access and eat for 1 hour, and then we sacrificed these mice and compared the fasting and feeding response. What we saw, as you can see in the healthy mice, if you look at the PBS, when we were allowed those fasted mice to eat, we saw elevation of phospho-AKT 308. We saw a suppression of phospho-AMPK and an induction of protein synthesis measured by pyromyosin incorporation. However, when we looked at the tumor-bearing mice, we did see that this feeding response was largely retained, although slightly muted, significantly muted, in the tumor-bearing mouse. But what we really were interested in is the fasting suppression of phospho-AKT, the accelerated activation of AMPK, and the decrease in protein synthesis after a brief fast in the tumor-bearing mice that was far exceeded what you would see in the healthy mice. So we went in, as we've been interested in IL-6 signaling in the IL-6 family of cytokines, including LIF, through the GP130 receptor in muscle that activates conical STAT3 signaling, and examine that role. And we're building off that in the LLC model. Melissa Pupp in my lab has shown that muscle-specific knockout of the GP130 receptor causes a reduction in gastroc mass, or less of a reduction in gastroc mass in the knockout animals compared to the tumor-bearing mice with GP130. When we did this with the tamoxifen-induced muscle-specific knockout of the GP130, and we followed the LLC model with the fasting paradigm, and we could see in the wild-type GP130 mice, given LLC tumor cells, an accelerated fasting of phospho-AMPK I've described to you, and the suppression of protein synthesis. However, in the knockout animals, the elevation of AMPK was normalized, showing that GP130 signaling potentially has a role in this elevated AMPK response, and that muscle protein synthesis was not rescued by the loss of GP130, causing us to now look more at the catabolic aspects of GP130 signaling on these responses. So Dennis Fix in my lab then went in to look, what about increased physical activity? And we again used the APC-Min mouse. And after the development of tumor burden, which are around 12 to 13 weeks of age, we allowed the mice again access to wheels for 4 weeks. And then we pulled their food and looked at the fasting response. What we were able to see is that even in this genetic model of cancer, that phospho-AMPK was accelerated in the muscle from 12-hour fast, and LC3B21 ratio was accelerated in the muscle beads, and we have other markers that go with that. But mice that were allowed access to wheels and also still had tumors, did not have this exaggerated fasting response. So we've gone on to look at the cancer environment, the muscle's response to repeated contraction. Justin Hardy did a lot of this work building up the Barr and Esser model of eccentric contractions and showing that we have an altered response or a blunted response with eccentric contractions. When we took that, and we've done some work also with low frequency contractions for 30 minutes, and Brittany looked at that in the fasted and fed state. And what we saw in the healthy, we were happy to see because I've wanted to investigate this for a while, is that in our fed mice, we see an elevation of AKT 308, but 3 hours post-contraction, no real change by contraction, but both indicators of mTOR activity through RPS6, phosphorylation, and muscle protein synthesis through puromyosin were synergistically activated in the fed healthy mice given protein, given contraction. However, when we look at the LLC mouse, we can see that the feeding response is there with the activation of phospho-AKT. But if you look over at the puromyosin plot, you'll see that there's no synergistic activation of contraction in the fed state on either mTOR activation or puromyosin. So we're very interested on how the cancer environment inhibits the synergistic effects of low frequency and feeding. So in summary, disrupted diurnal metabolic flexibility and cage activity occur in preclinical models of Maya. And decreased cage activity occurs before weight loss, and it's associated with later development of cachexia. And really the question we have is decreased physical activity and the muscle's altered response to contractile activity and feeding a driver of this early systemic metabolism disruption at the onset of cachexia. So in the closing minutes here, I'd like to just talk to you a little bit about the role for cancer treatment and long-term muscle dysfunction. We've been conducting some NCI-funded studies on the long-term effects of chemotherapy that are needed, because there's some very elegant work on the acute toxicities of chemotherapy on skeletal muscle. And so this premise for this is that 1 year after diagnosis and treatment, 67% of CRC patients report fatigue as a major factor. Many CRC colorectal cancer patients after treatment and report difficulties in the inability to perform daily tasks. And these are significant problems for cancer survivors. So we began to examine 5 FU-based therapies, 2 that have been well described by some elegant work by Andre Beneto's lab, include FOLFOX and FOLFIRI, which are a physiological cocktail used to treat colon cancer, and these components are used to treat other cancers too. And what has been shown is they can suppress acutely mitochondrial biogenesis, increase proapoptotic signaling, and actually FOLFIRI can create a molecular signature in muscle that's very similar to cachexia induced by colon 26 cells. So Jessica Halley at my labs worked in conjunction with Brittany Counts, worked out a paradigm that led to 4 cycles of FOLFOX chemotherapy. And then we looked at 10 weeks of recovery also after the completion of chemotherapy. And we did physical function markers. And what we saw is probably to be expected. FOLFOX chemotherapy, 4 cycles, decreased run time on the treadmill, decreased grip strength. And interestingly, though, after 10 weeks of discontinuing therapy, these weren't recovered. And we saw alterations to body mass, but not in lean mass. There was no loss of lean mass. This wasn't a cachectic type model, but we saw decreased fat mass both acutely, and the fat mass did not recover at 10 weeks after the cessation of chemotherapy. Corresponding to the alterations in body composition, we saw that 4 cycles of chemotherapy could alter carbohydrate oxidation and lipid oxidation when we kept these mice in the clams metabolic cages. Okay, I'll try to summarize here, but I just have a couple of slides. We also saw that 4 cycles of chemotherapy decreased COX-4 protein expression in both red and white gastroc muscle, BDAC protein expression, and OXPHOS complexes. So in conclusion, we then went in to look at the effect of exercise duration on some of these initial recovery points from chemotherapy. Exercise has been used during chemotherapy. It's difficult, but we set out our design to look at a higher duration of exercise, which corresponded to about 40%, a moderate intensity exercise that we've used for years. But we also tried a very low amount of exercise, 15 minutes a day, which only corresponded to about 10% of maximal exercises. And what we can see, whether it's 60 or 15 minutes, we were able to rescue run time at 4 weeks post-chemotherapy. And remember, without exercise, at 10 weeks post-chemotherapy, we still saw deficits in this. Interestingly, neither exercise training had any effect on relative grip strength, and it was not rescued. And so this was even though our 60 minutes a day treadmill exercise caused a significant increase in lean mass. So in summary, our results align with the growing body of research suggesting chemotherapy toxicity extends after the end of treatment, can affect whole body function, systemic metabolism, and some aspects of muscle biochemistry. So our question is there a role for inactivity in these processes, and are multimodal interventions involving exercise warranted post-treatment? And you can see here, we summarized the long-term deficits in runtime on a treadmill and grip strength that are independent of muscle mass. And interestingly, mitochondrial protein expression, while not recovering immediately and recovering faster in red muscle than white muscle, it was back to normal on our long-term recovery time point. So with that, I'd like to thank the organizers again and I'm willing to take any questions you may have. Thank you.
Thanks so much, Jim. We do have a question.
Can you hear me? Yeah, perfect. Elegant talk. Thanks so much. Quick question with regards to your last study there, where you noticed that there was not a loss in lean body mass post-treatment or your cycles, but there was a weight loss. Did you monitor food intake? Because one of the big things that occurs post-treatment is a lack of caloric intake in these patients, and sometimes they lack nutritional support.
Do you guys have Jim still? You don't have Jim still?
Oh, what a great question. Gee.
What? He asked for questions. I thought he was there.
Okay, wow.
That's an email question. Huh? It's not for me. Apologies. I thought initially it was recorded, but then he sold us on that one anyway. Okay. We're doing good for time here, so let me hand off to our next speaker, Mike.
Hey, good morning, everybody. My name is Mike Lisio. I'm from the University of Ottawa. I'd like to thank Dave and the rest of the organizing committee for putting on a great meeting and for giving me an opportunity to talk about some of the work we've been doing in my lab related to the long-term late effects of cancer therapy. And we know that in Canada, it's predicted that nearly 1 in 2 Canadians will be diagnosed with cancer across their lifespan, and this number is expected to go up. But the good news is that about 65% of patients diagnosed with cancer will survive beyond 5 years. And this is true for most of the countries that are represented here at this meeting. So in North America, Europe, and Australia, they all have very, very high cancer survival rates, which creates which is good news, but creates an issue of the long-term effects of cancer and its related therapies in people who survive. And these long-term late effects are particularly prevalent in juvenile or young adult cancer survivors because of the long lifespan after their treatment, and also because of the exposure to therapy during a time where there's rapid tissue development. And so these are some data looking at looking at markers of frailty in adults age 18 to 30 years old who are at least 10 years beyond cancer diagnosis. And we can see in adults at 18 and 30, at least 40 to 20% of these adults would be considered frail with at least 2 components of the frailty markers, which include loss of muscle mass, loss of muscle strength, decreased endurance capacity. And to put it into some context here, this level of frailty is similar to what a 65-year-old adult would experience. Okay? So you can consider this a model of accelerated aging. Looking more specifically at muscle in juvenile cancer survivors, again, this is long-term after their survivorship. About 80% of survivors experience some degree of atrophy and some degree of fibrosis, and this translates, like we heard already in previous talks, to significant impairments in mobility, which leads to decreases in quality of life. So mechanistically, what are some of the reasons responsible for this? And Jack Calaco's lab over the past few years has done some really nice work in this area. And he's shown that radiation exposure in juvenile mice depletes muscle stem cells. This depletes them, impairs their function. This inhibits long-term regeneration of muscle and also impairs muscle growth throughout the lifespan. So one might consider that just replacing these lost cells would be an easy solution to the problem. This is work from Andrew Brack's lab. So on the left, we had DS-red labeled satellite cells injected into regenerating muscle, and we see a nice recovery of the muscle fibers with the satellite cells incorporating into the mature regenerated muscle fibers 30 days after the regeneration insult. When we irradiate the muscle beforehand and when they irradiated the muscle beforehand and injected non-irradiated satellite cells, we can see that regeneration is completely blunted. The novel discovery in this paper was of a new population of radiation-resistant satellite cells that were tagged with or isolated by MX1 positive, When they inject these radiation-resistant satellite cells into non-irradiated muscle, you see very good recovery and regeneration of the muscle fibers with the satellite cells incorporating into the fibers, dyeing them green. However, when you inject these radiation-resistant satellite cells into a radiated muscle, you see significantly impaired regeneration of the muscle and impaired incorporation of the satellite cells into the muscle fibers. And so when we saw this paper in the lab, we thought that there might be something going on in the muscle environment that's impairing the function of these injected satellite cells as they are supposedly radiation resistant. And so if you're at the stem cell talks yesterday morning, you probably heard a lot about the different stem cell and other cell populations resident within muscle. You heard a lot about muscle satellite cells, pericytes, and endothelial cells. And in our lab, we're most interested in the fibroadipogenic progenitors. These were kind of first characterized and discovered in 2010 by two groups simultaneously. We're interested in these because in pathological conditions, they are the precursor cells that form adipocytes within muscle. They also produce a high amounts of collagen and contribute to ECM and fibrotic conditions in muscle. And some pathologies, they actually also form bone in certain conditions. We also know from other work that they are necessary for muscle maintenance throughout the lifespan, and they're also necessary for proper muscle regeneration. And so our lab's been looking at the response of FAPs to exercise for a while now. And so we've heard a lot about exercise so far, and in the context of exercise or of cancer survivorship, exercise is recommended by a lot of different associations. These are the recommendations from the American College of Sports Medicine. You can see that they recommend both aerobic, resistance only, or both combined aerobic and resistance training. And we see a lot of improvements long-term in a lot of outcomes related to quality of life in terms of physical function, some psychosocial outcomes, also fatigue that we've heard about as a problem in cancer survivors. We know a lot less about the mechanisms of how this works, how is exercise beneficial in long-term cancer survivors. And we also know that a lot of these data here are generated from adult survivors of prostate, breast, and colorectal cancers without much information on survivors of juvenile cancers. So in our lab, we tried to start to fill this gap in research. We had two graduate students who were very interested in this question, Nico Kalau and Olivia Sanders. And so we kind of combined two models that were available to us in the literature. We got very young mice, and we injected them with a rhabdomyosarcoma cell line that we got as a very generous gift from Crystal McCall's lab at Stanford. We inject this model directly into these cells directly into the gastrocnemius. The contralateral limb is injected with saline control, so it's completely within design. We let the cells engraft and grow for about 3 days, and then we treat the cancer similar to how it would be treated in kids, where we give them a single dose of vincristine chemotherapy IP, so it's a systemic therapy. And then after a couple days, we treat them with a course of fractionated radiation, so we give them about 5 gray per day over 5 days, and we just do that to the RMS injected limb, and we shield the rest of the mouse, so the rest of the mouse is not affected. After this treatment was done, we split the mice up into 2 groups. One remained sedentary with no access to a running wheel, and then we used the POWER protocol, or resistance endurance training protocol, that Charlotte Peterson's group with Kevin Murak spoke about yesterday very nicely. And so for those of you that weren't at that talk, what we do is we give the mice access to a running wheel, and we put magnets, each weighing 1 gram, unequally balanced on the wheels, so that every time the mice jump on the wheels and they want to start running, they have to overcome the inertia of the weight, and it provides a resistance exercise stimulus. It's a progressive protocol, so we start with 2 grams of weight on the wheel, and it progresses over the weeks till we reach about 6 grams, and then the mice stay at 6 grams for the last 3 weeks of the training intervention. Once the intervention was over, we collected the tissues, we collected the gastroc, and did some analysis. So during the intervention, we monitored how much the mice ran on the wheel, and they run between about 4 to 7 kilometers per day, and that stays, there's some variability there, but it's not significantly different. It's pretty stable across the time course of the intervention. We tested endurance performance in the mice. We tested it before they started their cancer injection and treatment, after the treatment, but before the resistance exercise protocol, and then after the resistance exercise protocol. And we found that the treatment, induced a decrease, but a non-significant decrease in endurance performance on the treadmill. And this was increased in the ret mice, the exercise mice, but maintained pretty flat in the mice that were sedentary. We looked at body composition as well using echo MRI at the same time points, and we see again a slight decrease but non-significant in percentage of body fat after the treatment. Body fat was increased in the sedentary mice but maintained low in the resistance-trained mice. Looking a little bit closer at the muscle, we removed the gastroc and weighed them. We saw a significant decrease in the gastrocnemius weight with the treatment and therapy, which was increased independent of therapy with resistance exercise training. Looking at muscle fibrosis, we did trichrome stains. We saw a significant increase in trichrome staining or markers of fibrosis in the treatment group with no difference really with exercise training. There's a lot of literature out there though that suggests that exercise training does remodel the ECM to make it more resistant, I guess, to be able to contract and adapt to the force changes. So we weren't totally surprised by this, but again, we did see the fibrosis that's common in cancer survivors with our treatment plus cancer group. Looking a bit more granularly at the muscle fibers, we did a fiber type stain and looked at differences in cross-sectional area. In the type 2A fibers, we see significant increases in the exercise train groups independent of the treatment and therapy. The type 2B fibers were significantly reduced in the treatment plus, or the cancer plus therapy group, but significantly increased in the exercise train group. And we didn't see really any effects in the type 2X fibers, maybe a trend with, for an increase in cross-sectional area with exercise training. And these adaptations are very similar to what Charlotte Peterson's lab has shown in aged mice undergoing a similar resistance exercise training protocol. We looked at fiber type proportion, and this was kind of interesting because the cancer plus therapy induced similar changes as the exercise did. So with both exercise and with the cancer plus therapy, we saw an increase in the proportion of type 2A fibers and a decrease in the proportion of the type 2B fibers. We're thinking that with the cancer plus therapy, the reduction in cross-sectional area and proportion of type 2B fibers is probably contributing to the reduction in strength that most cancer survivors see long-term. Looking at myonuclear addition, we didn't see any effects of exercise or therapy on, or cancer plus therapy on myonuclear number within the fibers. And so this led us to believe that perhaps there was some sort of satellite cell defect induced by the therapy and perhaps also by the systemic chemotherapy that was injected in the control mice that never experienced radiation or the cancer itself. So to follow that up, we did some flow cytometry to look at some different mononuclear cell populations in the gastrocs of these mice. We saw that the cancer plus therapy increased the number of hematopoietic cells in the muscles. We also saw that the resistance exercise training actually increased the number of endothelial cells in the muscle by flow, and this was actually trended to be reduced in the cancer plus therapy group. When we looked at satellite cells using alpha-7 as our marker, we saw that the cancer plus therapy significantly reduced the number of satellite cells per milligram muscle in the gastroc, and there was a trend for a recovery in the exercise train group. Lastly, we looked at FAPS here, and we saw again a trend for a decrease with cancer plus therapy and a significant increase in the number of FAPS after resistance exercise training by flow. So then we wanted to understand a little bit more about what kind of signaling mechanisms were happening inside of the muscles to try to get a bit at the mechanisms here. And so we did a gene expression array analysis of about 800 genes that are involved in inflammatory and fibrotic signaling, because it's been shown before in this model that the RMS plus therapy induces long-term inflammation and fibrosis. And when we did the analysis, we see when we cluster the samples together, there's a distinct distinction between the mice that got cancer plus therapy and the mice that were in the control, and a slight shifting with the exercise training, but nothing too dramatic. We performed a cell profiler analysis to try to understand which immune cell populations were enriched in the muscle, and that cell profiler analysis based on gene expression in the cells suggested that the number of macrophages was actually increased in the cancer plus therapy group with no effect of the exercise training. When we looked at gene expression and we wanted to understand better the effects of the RMS. So we compared the RMS plus therapy group that was sedentary to the control sedentary group. And what we found was pretty interesting in that almost every gene set that we looked at in the RMS plus therapy group was significantly upregulated. So these are the heat maps here, Red is an upregulation, blue is a downregulation. On this side, you have the RMS SED groups. On this side, you have the control SED groups. So you can see that everything was upregulated. When we looked at the top 10 enriched gene sets, we see that they're all related to macrophage activation, neutrophil degranulation, and we also see some effects here related to fibrotic signaling here with PDGF signaling, which is known to induce fibrosis. Looking at biological processes here, again, we see increases in the immune response and defense response, which is kind of consistent with what's already been shown in this model. So now, what are the effects of exercise in this model? So we compared the RMS RET group to the RMS SED group, and we found that 3 genes were downregulated, significantly downregulated in the RMS RET group. These were all related to immune cell recruitment. So we have CCL2, which is a ligand that recruits peripheral monocytes to the muscle. We have MyD88, which is involved in inflammatory signaling, and CD163, which is a monocyte, a macrophage marker, all downregulated in this model with resistance endurance training. We found 3 genes that were upregulated. These 2 right here haven't been extremely well described in muscle, but this PTGER4 is the gene for the prostaglandin 2 receptor, which has been shown before by Helen Blau's lab to be necessary for proper muscle regeneration. So we're excited to see an upregulation there. When we looked at the gene set analysis here, we saw that about 60% of the gene sets that were upregulated with the RMS plus therapy were downregulated when you add exercise, so a partial reversal of the gene expression. And these are the top 10 gene sets that were downregulated. Again, most of these were related to collagen biosynthesis, ECM synthesis, PDGF signaling, which is all related to maybe the fibrosis phenotype, which we couldn't detect in our trichrome, but we're seeing here at the level of gene expression perhaps. So to summarize that little study there, we're seeing that in this model of juvenile cancer plus therapy, resistance endurance training is improving performance and body composition, partially restoring some of the cellular dynamics in muscle, and also reducing fibrotic and inflammatory gene expression while potentially increasing some regenerative genes. And we wanted to follow this up a little bit more. Nico in my lab is very interested in the FAPs, and so he wanted to look more specifically at what the effects of radiation are on FAPs. And so I'll show a few slides here that from the work that he's been doing kind of independently, where he has, again, we have our model here where this time we just expose the one hind limb of a mouse to radiation, where we shield the rest of the limb. So we have the radiated limb and the contralateral limb as the control. We collect the muscles at different time points now after exposure to radiation. And we wanted to look at the different FAP subpopulations. Anybody here who's worked with some mesenchymal cells in culture knows that they're quite heterogeneous. And some recent single-cell RNA-seq data in the literature has suggested that there's a population of CD142 positive FAPs that are pro-fibrotic and a population of CD55 positive FAPs that are kind of more pro-regenerative. So we quantified these populations by flow cytometry at these different time points after radiation. We didn't see anything really happening in the regenerative population of FAPs. However, when we looked at the pro-fibrotic population of FAPs at 56 days or 8 weeks after radiation, we saw a significant increase in the proportion of this population of the overall total FAP population. So this is interesting phenotypic data, but we wanted to see kind of more functionally if this was actually the case. So Nico isolated some primary FAPs from skeletal muscle, cultured them, and exposed them to radiation in vitro. And we looked at fibrotic kind of differentiation, we'll call it, in a number of different ways. We stained for alpha smooth muscle actin, which is kind of a marker of an activated smooth muscle cell or a more pro-fibrotic marker. And we found that all of the cells, whether they were control cells, non-irradiated, irradiated, or a positive control where we had TGF-beta, which is a known inducer of fibrosis in FAPs, they were all positive for, most of them were positive for SMA. So we looked at the cell size, and cell size has been used as an indicator of fibrosis. You can see quite clearly the TGF-beta treated has an enlarged cell size. This is slightly smaller, but still much larger in the irradiated FAPs, and then smallest in the control FAPs. When we quantify these data, there's significant increases in cell size from irradiated to control. Another thing we looked at is collagen stress fibers. So again, Nico did these stains, and you can see the collagen stress fibers in the control group versus the IR group, where you can see significantly more of these collagen stress fibers. And then when you add TGF-beta, there's a further enhancement in our positive control. Quantify the data, significant increase in the irradiated group and the number of collagen stress fibers per cell. We looked at phospho-SMAD3, which is a known fibrotic signaling pathway. Again, this is increased in the irradiated cells compared to the control, further increase in our positive control. And then we looked at the amount of PDGF receptor alpha. Some models are showing that PDGF receptor alpha on FAPS is actually decreased as they progress to kind of a more myelofibroblast phenotype. And we see that both TGF beta and irradiation decreases the level of expression of PDGF or alpha in our isolated FAPS. How do we explain this? And so a study came out from an Italian group in 2020 that suggested that metabolism can direct FAP fate. And what they found in a model of in MDX mice, which is another model of muscle fibrosis and inflammation, that FAPs isolated from MDX mice primarily rely on glycolysis. So we looked at metabolism in our irradiated FAPs. Well, the first thing we looked at was mitochondria. And we can see in our FAPs under control conditions, not irradiated, we used a mitochondrial dye, and we can see nice reticular mitochondria, which is what you would see in a healthy condition. When we look at the irradiated FAPs, our mitochondria start to become more fragmented, which is an indicator of some mitochondrial dysfunction. When we quantified the number of mitochondria, interestingly, we saw that irradiation caused an increase in mitochondrial number, and we think this might be a bit of a compensatory mechanism here. So when we moved on here and did just a reminder of the increase in mitochondrial content in the irradiated group, this is 24 hours after in vitro irradiation of FAPs. We performed seahorse analysis, and we saw that there was no difference in the oxygen consumption rate. ATP production was the same between control and irradiated. However, there was a reduction in proton leak in the irradiated FAPs. So how we interpret this is that the increase in the number of mitochondria is a compensatory mechanism to maintain oxidative respiration, maintain ATP production, and the reduction in proton leak is driving more of the protons through ATP synthase as opposed to uncoupling proteins can try to maintain and maintain ATP generation inside the irradiated FAPs. When we looked at the glycolytic response in this, we looked at the extracellular acidification rate as a measure, a marker, I guess, of glycolysis. We saw that glycolytic capacity was significantly increased in the irradiated FAPs, and we looked at the ratio of the oxidative rate to the glycolytic rate, and that ratio favors glycolysis in the, trends to favor it at baseline and favors at max in the irradiated mice. All of these suggesting that perhaps the radiation is doing some damage to mitochondria, which is shifting metabolism to more glycolysis, and that might be involved in the myelofibroblastic transformation of the FAPs. Lastly, like I said before, FAPs are involved in regulating myoblasts through paracrine mechanisms. So we isolated FAPs from in vivo irradiated or control mice. We cultured them, collected the conditioned media, applied that conditioned media to myoblasts from a primary myoblast isolated from non-irradiated mice under differentiation conditions, and we looked at differentiation of the myoblasts. Here's the myosin heavy chain staining. This is the myoblast treated in differentiation conditions with control FAPs. You can see nice myotube formation, but when you treat the myoblast with conditioned media from the irradiated FAPs, you see a significant decline in the number of myotubes that are formed in culture. When we quantify that, when FAPs are isolated from 56 days post-radiation, a significant impairment in differentiation index in the myoblasts in this conditioned media experiments, and with the fusion index, that's impaired as early as 3 days, but also persists when you isolate the FAPs 8 weeks after radiation exposure. To try to understand what some of the signaling, paracrine signaling mechanisms were involved in that, we did single-cell RNA sequencing from these mice. We identified the typical 15 populations that we see often in single-cell analyses from skeletal muscle. And then Nico did a lot of work on the bioinformatics on this. And he did a cell communication analysis here. We're on the left side here of the heat maps. We have the sending cells, so the cells kind of producing the ligand. And then on the bottom axis here, we have the cells producing the receptor. We can measure the change in the number of interactions between radiated and not. This is all 8 weeks after radiation. And we can look at the strength of the interactions. And what we were interested in here is that the FAPs had some of the biggest changes in the number and strength of the interactions. And overall, these interactions were significantly lower. So here's the relative values over here. So radiation tended to basically make FAPs stop talking to almost everybody except for maybe the macrophages, where there's an upregulation of cell-cell communication. So next steps are for Nico's going to try to dig into this data a little bit more thoroughly and try to figure out what pathways exactly are activated, and what paracrine factors might be responsible for some of these effects. So hopefully we'll have those data coming up soon. And so it's just to summarize the second half of the talk here. In FAPS, the effects of radiation are we think to enhance fibrotic differentiation, which is related to enhanced glycolysis, and also impair the secretome. So I have to thank all of my lab for all their hard work, but obviously the ones who I have to highlight are the ones involved in this project. So Nico and Liv are the ones who did a lot of the heavy lifting. My previous postdoc, Donna, started these projects. All of the other students in the lab, I've listed kind of their posters here, so please feel free to go check them out. Our collaborators, particularly in the Burrell lab, who helped us with all the mitochondrial assays, and then all of our funders. So thanks very much for your attention. Happy to take questions.
Okay, we have 5 minutes or more for questions.
Fantastic talk. So I'm not very familiar with this model, nor with a lot of the FAPs, so forgive me, but was the intent of the treatment curative, and then how much of these cells that are changing in this particular sarcoma are manifested by actually tumor growth instead of impacting the surrounding muscle?
Okay, so the intent of the treatment was curative. A lot of the comment feedbacks we were getting from grant proposals was like, you're radiating healthy tissue, and so how does the tumor affect it? So we had to find a model that we could inject into the muscle so that we could only irradiate the muscle, and so, but we still wanted to cure it. We did a pretty good job of curing it. We started out with 20 mice total. 4 of them had a recurrence of the cancer and died, or sorry, 3 of them had a recurrence, died during the intervention. And then when we isolated 2 others, we found evidence of tumors in the muscle, at sacrifice at 8 weeks later. So to answer your question, there was no evidence of disease or tumor in the mice when we performed our analysis 8 weeks, in the mice that we included in the data I showed after 8 weeks of the exercise intervention.
Awesome. So you feel pretty confident that there's so the macrophage changes aren't related to tumor progression, more causing myopathy.
Presence of the tumor, as confident as I can be. We didn't dissect microscopically for the markers of the tumor, but we were pretty confident we were able to get it out. We had, and the 2 that had the tumor still in the muscle, it was very obvious. Their flow plots were it was basically a mess. We couldn't identify any populations because the tumors were pretty clearly there. So
makes sense. Thank you.
Thank you.
Oh, thank you for your talk. So I was really interested that the increased rates of glycolysis were occurring. So I know high-fat diets and fastings are pretty popular when it comes to cancer treatments, but do you think a more high-carbohydrate approach could be more protective for skeletal muscles and tissues now with that evidence?
That's good. We have tried those experiments in vitro, and we have some data on those. We want to look at them a little bit more. I don't think they're ready for prime time yet. So we have some in vitro data that suggests that manipulating the glucose levels in the culture media can impact some of the outcomes that we're seeing. We haven't tried anything in vivo yet.
Okay, thank you.
No problem.
Hi, Dr. Lisio, great talk. Based on some of the results that you showed here, it looks like the FAPs might be senescent. Is that something that you've considered looking into or investigating?
Yeah, so in our single cell data, the first thing we try to do is try to find a gene signature of the response to radiation, and 24 hours after, the CDKN1A, the gene for P21, was upregulated in all 15 cell populations across the board. It was still higher at day 56, but it wasn't as pronounced as what we saw at 24 hours. So it's possible that some of the cells that are there could be senescent for sure. Yeah.
Thank you.
No problem.
Just one question, sorry, before you jump off the stage. So in your first study that you showed, have you been able to do any muscle function readouts?
Yeah, we haven't. We did grip strength, but I don't think it was really, with a within design, one leg's radiated, the other one's not. It wasn't really a great outcome. We didn't see anything with grip strength, but I don't think it was the right one to do. If we repeat this, definitely would like to do some muscle function stuff. So we have the capacity to do it at Ottawa. We just didn't do it for the first study. Yeah.
Great, thank you. Okay, thanks, Mike. Okay, our final speaker in this session is Dr. Erin Talbert from University of Iowa.
Thanks so much to the organizers for the opportunity. It's my first IBEC meeting, but I don't think it'll be my last. It's been a really fun 4 days of seeing some people that I really like a lot and meeting some new friends. So the perk of going fourth in a session when you have 3 people who are really powerhouses in the field go before you is that you get to have a lot of fun. And so Andy says he only does work if he thinks he can get it funded or if he is funded. I like to do things because they're fun, which is probably an assistant professor mistake amongst many assistant professor mistakes that I'm making. And so I thought it would be a little bit fun to look through the data and what we know about muscle wasting and muscle mass in patients with cancer. And so we'll do that for a little while, and then we'll talk a little bit about some mechanistic things in ways in which I think exercise probably is beneficial for pancreatic cancer patients. And so we've talked a lot today about muscle mass in cancer patients, but my primary home is in a cancer center, and so I have to start by justifying my existence in the cancer center on a regular basis and talk about why we care about skeletal muscle mass in patients with cancer. And so this is a really nice meta-analysis that came out last summer that looked at 81 different studies, and they compared overall survival or hazard ratio of death in patients across a variety of cancers. And what they found is that patients who had low skeletal muscle mass in any type of cancer in these 81 studies were more likely to have a fatality, or their mortality was increased. And so there's a variety of ways these studies measured muscle mass, but this probably shouldn't be particularly surprising because this is true just in people without cancer, right? Low skeletal muscle mass is associated with shorter survival. But we also know, hopefully, there we go, is that low skeletal muscle mass is associated with low patient quality of life. And so patients who have low skeletal muscle mass report difficulty in doing activities of daily living, a lower quality of life. And so this is a really nice meta-analysis. One of the nice things that came out of nobody being in their labs for a while was some new meta-analyses showing, that patients with low skeletal muscle mass have lower quality of life across a number of studies. And this meta-analysis actually also separated out the functional part of these quality of life surveys. And they also showed that patients with low muscle mass had decreased functional quality of life. And so they felt like they were less able to do their activities of daily living. And as Andy talked about, things like difficulty in showering without taking a break, activities, climbing stairs, those types of things are really difficult for patients with lots of different types of cancer. And so we generally call muscle wasting in cancer patients a condition called cancer cachexia. But until relatively recently, it was actually pretty challenging to measure muscle mass in patients with cancer repeatedly. And so we generally define cancer cachexia as weight loss in cancer patients. And the primary clinical definition of when we consider a patient to be cachectic is loss of 5% of their pre-illness body weight over the last 6 months. And so patients with cancer cachexia very commonly have anorexia or decreased drive to eat, often have decreased nutrition. But what we think one of the hallmark features of cancer cachexia is that even if you provide these patients with enough supplemental nutrition to meet their daily nutritional needs, they're still losing weight. And so the definition of cancer cachexia is that it includes skeletal muscle wasting with or without the loss of adipose tissue. Now, new data actually measuring muscle mass in cancer patients suggests that it's almost always with the loss of adipose tissue. But it's still pretty common to see studies that are stratifying patients based upon how much weight they've lost because some of the difficulties of measuring muscle mass in cancer patients. And so Andy did a really nice job telling you about why we care about cancer cachexia, weight loss, and muscle mass in cancer patients, but there's a few things that I want to highlight. First of all, patients who have weight loss and loss of muscle mass with cancer have lower treatment tolerance pretty much across the board. They have decreased or increased number of complications following surgery. They're hospitalized for longer. The recoveries take longer. They have more negative outcomes in response to radiation. They often don't complete their prescribed radiation. And the same is true for their chemotherapy. As I'm sure all of you know, for many individuals, skeletal muscle mass is their largest organ, and it's a big sink for chemotherapy. And so the pharmacokinetics and the pharmacodynamics of chemotherapy are very, very different in patients who have lost skeletal muscle mass or have low skeletal muscle mass. And this really results in a lot of adverse events that end in patients not getting their full course of treatment. And I want to point out that there's some really neat emerging data to suggest that patients who are losing muscle mass or have low muscle mass are very much less likely to be responsive to immune therapy. So if you've heard anything about the changes in cancer care in the last 5 years, it's been about the increase in immune oncology. So one of the hallmarks of cancer is that it's able to escape your immune system and your immune system's ability to recognize cancer cells. And so there's a lot of data coming out from several groups suggesting that patients who are losing weight and have low muscle mass or immune therapy is ineffective in those patients. And so it's a population of patients who aren't benefiting from what really is, our best treatment right now. And as Andy nicely told you, skeletal muscle mass and function losses in cancer patients may be responsible for 20 to 30% of all cancer deaths. And so you've already seen this slide from Andy, and he did a better job explaining it than I will. But what I do want to point out is that there's, in the prevalence of cancer cachexia and in the severity of cancer cachexia, we can kind of see a divide in the patients from cancers like prostate and breast cancer who tend to be, these types of cancers tend to be life-altering for patients, but they tend to be more bumps in the road. And so we have lots of survivors of prostate cancer and breast cancer, whereas the cancers that tend to have high cachexia, high prevalence and high weight loss are very often life-limiting. And these are often cancers that will result in fatalities of these patients. And I take the time to point this out because the vast majority of what we know about exercise in cancer patients and what we say exercise in cancer survivors comes from this side of the table. Okay? We know very little about exercise in patients who have cancers that tend to be diagnosed at an advanced stage and tend to limit the life of these patients. And so we'll talk a little bit about that. So much like the Judge Lab, my lab is interested in cancer in pancreatic ductal adenocarcinoma patients, or PDAC patients. Cachexia and weight loss is very prevalent in these patients, which makes it easy to write grants. But I also personally think that this is the population of patients who's most likely to benefit from an anti-cachectic therapy. And the reason I think that is that weight loss and muscle wasting in PDAC patients is not solely associated with late-stage disease. Okay? So in the previous slide that we both showed you, looking at the prevalence of cancer cachexia, weight loss and cancer cachexia is very prevalent in lots of patients who have end-stage metastatic disease with multi-system organ failure. But data across many institutions and labs suggests that perhaps 70% of pancreatic adenocarcinoma patients have lost at least 5% of their body weight at the time of their diagnosis. Okay? These patients have stage 1 and stage 2 disease, so operable disease. Unfortunately, the survival still is not excellent in patients who have stage 1 and stage 2 disease, but these patients still have theoretically curable cancer. And so I think that that's really a population of patients who has the ability to benefit from an anti-cachexia therapy. And so I wanted to take us time to point out that pancreatic cancer is a disease of the aged. So this is data from the US SEER database, but it's similar around the world. And so pancreatic adenocarcinoma is not particularly prevalent in individuals between the ages of 40 to 64. However, it's much more prevalent in individuals between the ages of 65 to 74, and even more prevalent in individuals that are above the age of 75. And as you can see, these trends that are increasing, pancreatic cancer is one of the only cancers that's actually increasing in incidence. These increases are generally occurring in older individuals. And so what this means is that cancer cachexia and muscle wasting in cancer patients is being compounded by age-related sarcopenia. And I think that it's really important that we keep in mind that many of the things that we're looking at and the questions we're mechanistically interested in are happening in the context of sarcopenia. And as Jim pointed out earlier, also probably happening in the context of inactivity or disuse. And so I think we really have an interaction effect between age, cancer, and inactivity, which is really contributing to these functional problems in these patients. And so the median age of a pancreatic cancer diagnosis in the US is 71 years, and it's very similar to around the world. And as Andy nicely pointed out, this is having an impact on the way that we think about preclinical animal models and trying to do mechanistic studies. There are many studies showing that similar cachexia occurs in young and old mice. Sometimes the tumors grow a little bit faster in young mice. But I think that it's definitely something that a lot of us are spending time thinking about and making sure that we're really capturing the interaction between age, cancer, and inactivity. So what causes muscle wasting in cancer patients? And so for 40 years, the assumption has been that there's some circulating factor that's coming out of the tumor. And so in addition to effects on muscle mass, we know tumors also have effects on adipose tissue. Those effects on adipose tissue may make losses of muscle mass more likely. And we also know that tumors secrete factors that have impacts on the central nervous system and the brain, those anorexia effects that we talked about earlier. And so I would say that the strongest evidence that we have that a secreted factor coming from the tumor is responsible for muscle wasting in cancer patients is the fact that when patients' tumors are effectively treated, the weight loss stops, right? So when cancer has gotten back under control, the weight loss in these patients stops, the muscle wasting stops. They often are able to put on some adipose tissue, but often don't add skeletal muscle mass. And so based on cross-sectional human studies, looking at groups of patients at a single cross-sectional time point in animal models, these 4 circulating factors have been identified as potentially causative for cancer cachexia. Okay, so interleukin-6 or IL-6, tumor necrosis factor, TNF, activin A, and myostatin, which is also called GDF15, have all been associated with cancer cachexia and weight loss in cancer patients. Unfortunately, all of these circulating factors have been targeted in clinical trials, a variety of phases of clinical trials, and all those clinical trials have failed. And so while it is absolutely possible that any of these circulating factors may be responsible for muscle wasting and weight loss in cancer patients, just targeting one of these factors in a blanket group of patients is not turning into a successful anti-cachexia treatment. And we think that that's probably true because we're really starting to develop an understanding that cachexia in one patient doesn't look like cachexia in perhaps all other patients. And so I'm sure that you've all heard about the revolution in personalized medicine, and we call it personalized oncology in the cancer space. And I think there's a lot of emerging evidence to suggest that perhaps that's something that we need to be thinking about in cancer patients when we're worried about their cachexia and we're thinking about why they're losing muscle weight. And so this is just a heat map of a number of different patients and their circulating levels of different cytokines or circulating factors. And so over here, we have control patients who do not have cancer. And so red, or whatever this color looks like to you, is high. Blue, or whatever this color looks like to you, is low. And each column is an individual patient. And so you can see there's some variability in the controls, but for the most part, they have pretty low levels of these circulating factors, as you would expect from healthy people. When we look at patients who do not have weight loss, so these patients have pancreatic ductal adenocarcinoma, but they do not have weight loss, for the most part, a fair amount of blue, but some specklings of red, of increased levels of these circulating factors, which are things like IL-6, activin A, myostatin, GDF15, which is a circulating factor that people are really interested right now in cancer cachexia, and then MCP1. And so that you can see in these patients over here with pancreatic ductal adenocarcinoma who've lost at least 5% of their body weight, you can see some red, However, it's definitely not a blanket across the board elevation in all of these factors. And so a patient like here might not benefit from an anti-cytokine therapy from any of these circulating factors and might need a more personalized medicine, where a patient who has high IL-6, like this one, or high inflammatory burden might benefit from a treatment targeting those circulating factors. And so I think that this is something that we're starting to appreciate, is that there's probably not a one-size-fits-all solution here. And so because we've had so little success in clinical trials in cancer patients for cachexia, people have started pursuing what we're calling multimodal interventions. Okay? And so most of these interventions include nutritional counseling or meeting with a dietitian, sometimes once, sometimes multiple times. They often include some sort of anti-inflammatory to try and tamp down markers of inflammation. And sometimes it's an NSAID, sometimes it's fish oil. And then they often involve some sort of prescription for exercise. So maybe, there we go. So Mike nicely showed you this slide talking about the American College of Sports Medicine's recommendations for exercise for cancer patients, both cancer survivors and people with active cancer. But it's pretty much the physical activity recommendations for the rest of us. But I like this slide because it really nicely shows where this evidence comes from. Okay? And so we know that there are 7 cancers that, from epidemiological evidence, are less likely to occur in people who exercise. Right? So these people are less likely to ever have cancer to start with. Secondarily, we know that survival is longer in 3 common cancers, right? So people who exercise survive longer when they're diagnosed with these cancers. And as you probably already suspect, the cancers that we have this data for are breast cancer, colon cancer, and prostate cancer. And there's a lot less data about what patients who have active cancer, and particularly those active cancers that are likely to limit their life. And so there's 4 professional organizations for medicine who have recommendations for cancer cachexia. 2 of them have no recommendation or no mention of exercise. And then the other 2 in 2021 guidelines again suggest that patients with cancer should be meeting physical activity guidelines for any adult. But again, they fully admit that they're based on very little evidence. We don't have phase 3 randomized controlled trials of patients with active cancer, like pancreatic cancer, like lung cancer, like hepatocellular carcinoma. And so the first thing I want to point out is that it's probably true that the goal of any intervention in these patients, whether it's pharmacological or it's exercise or any other intervention that you can come up with, nutritional, is probably to help these patients hang on to the muscle mass that they have. Okay? Probably building muscle mass in the context of cancer, particularly active cancer, and inactivity, and aging is probably a big ask. But I think there's enough evidence to suggest that patients with cancer are very likely to benefit from exercise. And I think we have a pretty good idea that patients with cancer are very likely to benefit, just like all of us who don't have active cancer tend to benefit from exercise. Okay? And I think patients with cancer are very likely to benefit from reduced inflammation associated with cancer. We know that the exercise alters the immune system. Cancer is definitely an inflammatory disease, and altering the immune system is very likely to benefit patients who exercise. In patients with cancer, we don't have really strong evidence that there is significant catabolism or there is significant decreases in anabolism. Okay? There are not that many studies done in human patients, and they're all done under different conditions. And so certainly for some patients, there is significant catabolism, but it's probably not true for all types of patients. Similarly, many patients probably have a decrease in protein synthesis. There's questions about whether this is a decreased response to feeding or just generalized decreased protein synthesis all the time. But again, patients who exercise are probably very likely to benefit from stimulating anabolism. And then the last thing that I'd like to spend a little bit of time today on thinking about what exercise might do for patients is promoting muscle regeneration and repair. And this is something my lab is particularly interested in and think that is a significant cause of muscle atrophy, particularly in patients with cancer. And so, as I said earlier, it's only relatively recently that we've had good data about muscle wasting in cancer patients and change in muscle mass in cancer patients. And so sometimes in science, you do the experiment you can do, not necessarily the perfect experiment that you'd like to do. And so we've been really lucky to have access to rectus abdominis muscle biopsies taken from patients with pancreatic cancer when these patients are undergoing an attempted resection of their tumor. Right? And so these patients are coming to the hospital, they're going to have an incision, and they consent to letting us have a little bit of muscle from that incision site. And part of the reason that we do it this way is because it's a very low ask of a cancer patient, right? Anytime you're thinking about doing something in humans, you have to think about the burden that you're putting on patients and what you're asking them to consent to. Cancer patients have a lot of stuff going on, and they don't necessarily want to make repeated trips to your lab to do vastus lateralis muscle biopsies. And so this has been a way that we've been able to get a fairly high consenting rate and have access to these tissues. I had not, when we started this experiment, really appreciated what the challenge would be of coming up with age-matched non-cancer controls. It's been a really interesting road to figure out what is a good control for an abdominal operation. And so the controls that I'll show you today are from patients who are undergoing reversal of a colostomy. Sometimes it's a lymph node transplantation to reduce lymphedema, but it's definitely been interesting to try and come up with what would be a good way to get rectus abdominis muscle from 70-year-old patients. And so Andy nicely showed you that there are very often significant structural changes in skeletal muscle of cancer patients. However, there's also a population of patients who lose significant weight and we believe muscle mass, but still have pretty functionally normal muscle, or at least when you look at a cross-section of these muscle fibers, it doesn't look too different. And so this is a control patient over here. Actually, that's not true. This is a patient with pancreatic cancer who has maintained their weight, and we've got these nice, beautiful muscle fibers. But even in patients who've lost 20 to 25% of their body weight, sometimes the muscle still looks phenotypically fairly normal. However, when we look closer, and particularly through electron microscopy, we can see that in a control patient, we have these very nice, smooth muscle membranes. But in patients who've lost weight and muscle mass, we have these messy, no longer smooth muscle membranes, and particularly in a patient with more severe muscle mass losses, you can see all these hatched-like fibers across the muscle membrane. And so we think that that seems to be a disorganization of collagen 1. And so there's a lot of collagen 1 in skeletal muscle surrounding individual muscle fibers. And we think that tumors are secreting some factor that's causing a disorganization and a breakdown of this collagen. And you can particularly see this increased staining brightness in patients who've lost a significant weight. And so one of the things that I'm pretty sure is true about patients with pancreatic ductal adenocarcinoma and cachexia is that they have an accumulation of muscle progenitor cells. And so if we take these muscle biopsies from cancer patients, we degrade away the muscle fibers and are left only with the muscle mononuclear cells. We can see that as a patient has increased muscle weight loss, there are more of these muscle stem cells as evidenced by being CD56 positive and also lineage negative. And so we think that this is representative of a regeneration deficit in skeletal muscle. of cancer patients. And so, as I'm sure you're all familiar, when muscle is damaged, it goes through a process of activating muscle progenitor cells that then proliferate, differentiate, and fuse in to repair the muscle fibers. And in cancer patients, this process seems to be broken. And it seems to be broken somewhere in here in the switch from a proliferative phenotype onto a differentiation phenotype. And so we think that this disorganization of the extracellular matrix may really be contributing to keeping these muscle progenitor cells in this proliferation phase. And we think that this inability of muscle progenitor cells to differentiate really is an environment problem, because if you do those same mononuclear cell preps and you take the muscle mononuclear cells out of a mouse with cancer who's lost muscle mass, and you just plate them on a dish in tissue culture, they actually differentiate better than cells that come from a control mouse. And so we think these cells are really primed to be able to differentiate. They really want to differentiate, but it's the environment surrounding them that is preventing them from doing so. And we think this is particularly true in super resolution photos. And so this is collagen 1 staining here in green, and then this PAX7 represents a muscle progenitor cell. This is the control patient without cancer. You can see these really beautiful collagen fibrils surrounding skeletal muscle fibers. However, when we look at a patient with cachexia, we actually don't seem to have the structure of those collagen fibrils anymore. It seems to have actually fallen apart, and you can actually see this muscle progenitor cell kind of is rounded up and kind of has what appears to be a more active phenotype for these types of muscle progenitor cells. And so my lab is really interested in trying to understand how in mice with cancer and patients with cancer, we go from here in this beautiful collagen structure to here where that structure has been disorganized. And we think that cancer is injuring muscle somehow, probably through a disorganization of the extracellular matrix, activating these muscle progenitor cells, and then the inability of these muscle progenitor cells to differentiate is leading to atrophy. And so we think that there's a lot of potential for anti-cachexia therapy to be able to stimulate these muscle progenitor cells to differentiate, to remove this block in differentiation, and that'll help us maintain muscle mass. And I certainly think that this is one way in which exercise might be beneficial to patients who have active cancer. And perhaps there's other ways to do this pharmacologically, but to be honest, exercise might honestly be the most efficient way to do this. And so I want to spend the last couple of minutes of my talk on a study done with my colleagues. And so one of the nice things about working in this space is that you develop clinical colleagues who are interested in your science and want to contribute. One of the problems of developing clinical colleagues is that they're not particularly patient. They tell you they have patients sitting in their office right now and they really could use a solution. So if the PhDs could hurry up it a little, they'd really appreciate it. And so this is an effort from two of my colleagues that I participated in, Carlos Chan, who's a surgical oncologist, Amy Perlman, who's a urologist, in an effort to help patients now. And so Amy asked Carlos, who asked me, if we had any idea about testosterone levels in patients with pancreatic cancer, and particularly male patients with pancreatic cancer. And what we found out is no, we really don't know. And so Amy was interested in this because both fatigue and weakness are symptoms that are common to both cancer, cachexia, and weight loss, and having low circulating levels of testosterone. And so we went back through the medical records of 1,500 male pancreatic cancer patients that had been treated over the last 20 years at our cancer center, and found out that only 2% of them had ever had a blood testosterone value taken. So that wasn't going to help us get any data. And so we actually went back to our tissue bank and pulled serum from 90 male pancreatic cancer patients. And as it turned out, half of these patients had what would be considered normal testosterone levels. Half of them had what would be considered low testosterone levels. And we didn't find any difference in the incidence of fatigue or weakness or performance status in any of these patients who had low testosterone. but that certainly tells us that there's a very high prevalence of low testosterone in these patients. And so Melinda Sheffield-Moore has done some work in this space, both in male and female patients, with small studies looking at the safety and feasibility of testosterone supplementation to improve the symptoms of cancer cachexia, and has some promising data, but the studies weren't really powered to be able to show effectiveness. And then there's a large VA-funded trial going on looking at testosterone supplementation. But what Carlos and Amy decided to do was actually a registry trial. So instead of waiting for a phase 3 big trial to tell us that testosterone supplementation is beneficial for cancer patients, they're actually doing it now because it fits within the scope of standard practice. And so every new patient that comes into Carlos's office gets asked if they want to have their testosterone screened. And if they do, they're actually referred to Amy for a testosterone screening and supplementation. And they're doing quality of life studies before and after, to be able to get some real world data of implementation. And then I want to make one more point talking about the presence of sexual dimorphisms in cancer cachexia. Jim, who we heard from earlier today, has really been a leader in thinking about whether male and female preclinical models show the same cancer cachexia, and Nick Green at Arkansas has also really led us in this space in preclinical models. But I want to point out that these dimorphisms are really now that we're looking more carefully showing up in patient data. And so these are just survival curves stratified by whether a patient has lost more than 5% of their body weight or not. And in males, you can see this nice separation. In females, you see no separation. And so this is only about an N of 40 in both males and females. And so maybe it's a quirk of the data, but maybe not. And Teresa Zimmer's recently published some data suggesting that muscle wasting and loss of muscle is actually very uncommon in females with cancer cachexia. And so this is a longer story for another day, but I think I'd be remiss if I didn't tell you that we're really seeing a significant expansion of this data and really appreciating that, while the incidence of pancreatic cancer is very similar, while the weight loss in pancreatic cancer patients is very similar, it's very possible that the phenotyping isn't. And Andy was kind enough to show some older RNA-seq data for me of pancreatic cancer patients and that downregulation of those genes. That's data in males. We did that exact same experiment in females at the same time, and we found 0 differentially regulated genes in muscle. And so that's something that my lab is actively working to understand. So if you take anything away from my talk, hopefully you'll appreciate that muscle wasting in cancer patients is probably an age cancer inactivity effect, and we need to think about that. Exercise is very likely beneficial for cancer patients, but unfortunately we're still waiting on foundational studies to show that, and you need to be making thoughtful considerations of sex in your studies. And so Carl, Mary, and Dave are the best clinical collaborators a postdoc could ever ask for, and really have allowed me to develop these ideas. This is my lab. Like everybody else, we're looking to add one more person to the lab. There's some flexibility in whether it's at the tech level or the postdoc level. So if you're looking for a place to come hang out and hopefully do some fun science, we'd be happy to chat. And with that, I'll be happy to take any questions.
Okay, floor's open for questions.
Thank you, great presentation. You looked at, or you talked about personal medicine also in relation to the pancreatic cancer, and you showed some differentially upregulated cytokines. I wondered whether insulin, plasma insulin shows a similar pattern depending on the patient, and whether that's also related to the weight loss you see in patients?
Yeah, so new onset diabetes is very commonly associated with new diagnoses of pancreas cancer. We have virtually no understanding of that in terms of cachexia. And so I think that it's an area that people are interested in, but we really don't. I can't think of any good data. Somebody correct me if I'm wrong, but I cannot think of any good known data. It's something that we're starting to dig into in some mouse models, and so we're hopeful that if we see that it might be causative of cachexia in mice, we'll go back to the patients, but I unfortunately don't have a satisfactory answer.
Thank you.
Sorry. Wish I did.
Great talk, thank you. Fran, University of Arkansas again. And I was wondering, you guys are looking into testosterone as a way to treat or maybe try to mitigate that cachexia in patients. And I was wondering, because we see so much of a protection in the females in muscle wasting and even in mitochondrial health, somewhat in muscle function as well. And I was wondering if you guys looked into estrogen levels across patients, and if you have any opinions or speculations you can do with maybe a protection side of estrogen in the females.
Yeah, it's a great question. So I don't have a satisfactory answer. What I can tell you is that all of these patients are postmenopausal, right? So it's a disease of the aged. And so we don't think that this difference that we see likely in muscle wasting and likely in differential gene expression is probably due to circulating estrogen. Now, whether it's due to the lifelong presence of estrogen, that's a different question. And so we have some experiments that we're starting in the lab to try and tease that out. But we don't think it's necessarily the fact that there is estrogen here now. And there's studies in mice of overoptimized mice that don't see much difference in muscle wasting between those. And so, as you know, there's lots of studies that show estrogen is protective for some things. We don't think necessarily in this case that's the source of our sexual dimorphism. But again, I think very possible that it's lifelong estrogen exposure, but not estrogen at this time.
Hey, nice talk.
Thanks.
So 2 questions. Do you think that in your model is differentiation that is blocker, or is the secretome, so in the niche interstitial cells, they start to secrete different kind of cytokines that now affect the muscle? So have you compared the secretome of the interstitial cells in a cancer cachexia versus a control, or not cachectic people? And my second question, it is true that there is a difference in men that are cachexia versus non-cachexia, and not in females. But if you look at survival of men, it's much longer, and men have more muscle mass than women. So have you tried to correlate this kind of how much is the lean mass compared to the survival more than just a weight loss?
Yeah. So your first question, not to the level that will be a satisfactory answer for you. We have done some Western blots for cytokines, and so you can find upregulation of IL-6 within the muscle. You can find a little bit of additional IL-1 beta within the muscle, not a lot of extra TNF, if I remember correctly, in the muscle. But no, we really don't have a good understanding if it's a local microenvironment or it's necessarily something coming from the outside. And so that's hopefully where we're headed next. For your second question, in the slide I showed of the 2 differential survival curves, the males and females had identical overall survival, right? So if you just put those 2 curves on top of each other, males and females, they're the same. And so I don't have a better elegant answer to your question. What I can tell you is we're doing now is we've actually gone back and we're retrospectively looking at a population of 200 pancreatic cancer patients, and we're actually following how their muscle mass changed on their CT scans over time. And we really have powered it to look at the dimorphism between males and females. And we're really trying to take into account survival. And we're also trying to take into account a response to treatment. And so I have a very brave biostatistician that I don't think knows quite what it is that she's signed up for, but she's actually attempting to model when treatment is effective, which is generally when we think patients are more able to maintain their muscle mass, and then model then when their treatments are less effective, and tease out the dimorphism that way. And so I hope about a year from now I'll have a more satisfactory answer for you. Thanks.
Are you looking at muscle quality in that same study, in addition to muscle mass from the scans?
We're trying. It's turned into then a more difficult problem than we thought for. So you can look, one of the things that's really changed in our field is the ability to use CT scans that have been taken for patient care over the course of their treatment, right? It's a lot to ask a cancer patient to come in for multiple DEXA scans. Even other methods of measuring muscle mass are a lot to ask a patient. And so you can look at the quality of muscle in those patients, looking at just the density of muscle, and you can also get some hints of adiposity. And so we're trying, but unfortunately the CT scans were taken on a lot of CT scanners at a lot of different places. And so there's some differences in the scans that we're having to, the model is getting bigger than I think my bioinformatician wants it to be. So hopefully we'll have, the data to be able to show those changes, but to be seen.
Great. Okay, we are out of time, so thank you again to all the speakers.